Hey, this is Joel Duff. Well, I guess you knew that since you clicked on the link, but or maybe you just looked at the title and you said, I just got to know what this thing is about the genetic code and codon bias. Sounds kind of like some geeky genetic stuff. Well, it is, but I'm going to try to explain the basic concepts very simply in order to try to answer some questions that came into my mailbox. So this is my mailbox, episode number 2. It's where I dip in and look at, hey, what if people asked me either in comments on my channel or on my blog or sent me emails? And this particular case, I'm responding to several questions from an email contributor. And so without further ado, let's get to it. I'm going to talk about common ancestry and the genetic code. How are those related? What does that mean? So let's get to it. Let's talk about common ancestry and the genetic code. And also, what about codon bias? Well, what is codon bias? We'll get to that in a few minutes. All that and more coming up. All right, so here's our first question. Some people would claim that the near universal genetic code is not evidence for common descent because the same intelligent designer would use the same code in different applications. In response to this, are there other differentiating lines of evidence that show that the genetic code is supportive of universal common ancestry? For example, are other genetic codes possible, with some being just as, if not more efficient than, the standard genetic code? Really fantastic question. It's going to take me just a little bit of background material to bring watchers up to speed as to what's being asked here. But I think this is an excellent question. The question is about how information is stored in living things and the code that we use, and the common argument that because the genetic code or the information system that living things use here on Earth is has a high degree of similarity between organisms, that is an argument for common origin or common descent, right? That there was an original organism that established the code of how we read our DNA, and then that was passed on from that particular organism to its descendants. And through its descendants eventually arise all the different forms of life, which then share that particular genetic code. In response to that, one could say maybe the reason that all organisms share a common genetic code is because they all have a common creator, right? If you had an intelligent designer, the God of the Bible, for example, who creates the world and all the living things in it, and says that in creating that, decides to use the same code, right? And or the same basic, you could think of it as the same basic operating system is going on in living things, and simply applies the code to different situations and different environments to create different programs, but still using the same operating language. All right, so in response to this, going back to the question, are there differentiating lines or other lines of evidence that show that the genetic code is supportive of universal common ancestry? And in particular, this reader is interested in whether other genetic codes are possible, right? Is this the only code that's possible, or is it the best code? Are there other equally valid codes that could have been chosen to be used either by an intelligent designer or by a selective process, evolutionary process? Could we have ended up with a different code, or is this simply just the best code we could imagine there would be? And you could either argue an intelligent designer used the very best code, or evolutionary processes generated or selected for the very best code that's possible. These are really intriguing questions for which I don't have like the answer, right? But I can provide you some background and give you some ideas about how to think about this particular question. I'll probably provide some personal perspective on it myself. So what is this genetic code thing? Let's talk about that for a moment. And I really like how the reader here, the person who provided the question, calls it the nearly universal genetic code or near universal genetic code. You may have grown up learning about, in your basic biology class, about the universal genetic code and learned how we read our code and turn it into products, which are typically proteins. And it might have been called the universal code, but now we know it's not actually universal, so we call it the nearly universal genetic code. But we'll get to that in a moment, and it's the very idea that it's not completely universal, which might give us some hints, give us some tips as to how to think about this question about what a designer would do and what evolutionary processes would do and what we'd expect from them. So what is the universal genetic code? It is a series of 64 sets of 64 combinations of 3 letters. Remember, your DNA has 4 possible letters that make up the rungs of the ladder, right? You can have A, T, C's, and G's, which are just shorthand for different chemicals: adenine, cytosine, thymine, and guanine. And so we usually talk about the A, C's, T's, and G's. And our genetic code is read in what are called triplets, or 3 base pair pieces. And those 3, if you were to arrange things of 4, right, A, T, C, and G, and ask how many combinations of 3 of those can you make, it's 64 of them. So you're seeing every possible one. First, you might be wondering, like, where's the T's, right? Because I said A, C, T, and G. But when DNA is read into the message that is going to be carried out to where it's going to be read into the language of proteins, it's turned into this thing called RNA, and specifically a messenger RNA. And in messenger RNA, all T's, thymines, are converted into uracils, which is what the U is for here. It's a very similar chemical. And so you have the code would tell you that if you have a UUU at a particular position in your genetic code and on your chromosomes, right, in your nucleus, that UUU would be read by the translational machinery, a large protein complex that says, how do I understand this genetic code and turn it into like physical products like these enzymes or different structural proteins? And UUU is turned into phenylalanine, which is an amino acid, all right, a different kind of chemical. UUA is turned into a leucine. Let me get my pen out here. So UUA is turned into leucine, but CUG is also turned into leucine, and AUU is turned into isoleucine. And so there's 20 different amino acids here that make up your proteins. Right? So when you have protein in your diet, you're really eating these amino acids, these 20 amino acids, some of which you're consuming in order to build more proteins for yourself. And this is the instruction code, the A's, U's, C's, and G's, and specifically the order in which they are placed in a gene, which is just a length of code that corresponds to or is read into a specific order or sequence of amino acids, which is a protein. All right, so you have 20 amino acids, you have these 64 codes, and we'll talk about redundancy in a moment, but if you only have 20 amino acids, there must be more than one code for many of the amino acids. So, I've been explaining this, what's called the central dogma of genetics. You have DNA, right, that you're storing in your nucleus. And then when you say like, hey, I need something, like I need to make some insulin, right? Well, you have stored inside of your genome, the information in your nucleus, you have stored away somewhere in there the code that tells you like, here's how you make insulin. And you got to go get it, all right? So you have a way of opening up your code and then making a copy of that code. That's what's called the messenger RNA. That's going to be the message that takes the information from your nucleus where you're storing it. Kind of think of it as like your hard drive, right? You have all the information for all the programs you're going to run on your hard drive. When you want to actually like use one of them, you go in and you actually copy pieces of that information out of the hard drive, put it in your random access memory so you can use it, you can compute it, right? You can execute those files. That's just what you're doing here in your cells right now. You're taking a sequence of DNA, you're converting it into a sequence of RNA, which has the U's in it, So it's U's, C's, G's, and A's. And you bring out that particular piece of code, all right, a piece of information, in this case, the information for how to make insulin. And then you have to translate the information. So you have what's called transcription, because you're simply transcribing genetic code into another form of genetic code called RNA, ribonucleic acid. And then you're taking your RNA, you're taking the message out of the nucleus, where you're safely keeping your original code, and you're taking this copy of your code out into the cytoplasm, the rest of the cell material, and there it will hook up with this large protein complex called a ribosome, which is responsible for translating, all right, reading the code. Now, how does it read it? Well, you have these little chemical units, tRNAs, that have 3 base pairs, 3 AU, C's, and G's, that complement the strand that you just had out there. And that's really what's happening here. You have these little units that identify, oh, you have a UUU right here, right? Let's say your code is UUU, and then you have, let's say, AAA. All right, let's write out some code. AAA, CC. I'm making it easy here for me to draw. Well, it's terrible because I can't draw, but you get the idea. Let's say we had a genetic code of UUU, AAA, CCC, GGG, and you were to ask, well, what does that become? Well, you would start reading your code, and as you read your code, you look at your first 3 base pairs and you say UUU. Oh, that's phenylalanine. All right, so that makes PHE. And then you say, well, AAA, what's AAA? And so your ribosome, your reader, is going to look for the corresponding amino acid that goes with AAA. We'll look down here. We look on our chart, right? Our code reader, which is what we're using, and we end up with lysine. And so we say, okay, for that one, we're gonna make lysine here, and then we have CCC. All right, so I have to find CC. Where is that? Right here. So we've got proline. CCC is going to make proline. And then we're going to end up here with GGG, which is glycine. All right, so we're going to have GLY, glycine. And then the ribosome looks at each one of these successive sets of 3 called codons, it's going to bring in the corresponding amino acid that goes with those. And then it's going to hook those amino acids together. And then release the mRNA again, the message again. And so what you're done with is you have a series of 4 amino acids that are hooked together in a certain order. And a particular order of amino acids is what makes a protein have the structure it has, right? My hair is kind of curly because it has a certain order of amino acids that when they wind up together, they form a curl, right? Because your hair is protein. Right, your insulin hormone is a proteinaceous thing, and so it has a code, right, for making the particular shape that it has, which has a certain function in your body. And so you have thousands of these particular things inside of you, and they're all encoded. Now, the person that asked me this question, I'm sure knows all this, but I'm just providing the rest of you with sort of this background information so we can kind of understand what this question is about. And so if looking big picture now, this 64 base pair, at least 64 different codes, is called the universal genetic code, or nearly universal genetic code. So this means that if I were to say, hey, this UUU, AAA, CCC, GGG, surely somewhere in you, I'm sure, you have that particular sequence maybe inside of a gene. And so when you read it, you're going to read those 4 amino acids. But I could take this sequence out of you and I could stick it into a bacteria, and I could ask the bacteria, right, to read that code, and it would read it as phenylalanine, lysine, proline, glycine, exactly the same way that it does inside of your cell and my cell. And I could take that same sequence and put it into a fungus, and I could take that sequence and put it into a plant, right? You get the idea, right? It's all the different organisms, well, 99.99% of all the species of all organisms, of the millions of different species on Earth, read the same code. And this is what allows us to move genes from one organism to another. And enabling other organisms to read those codes is because they use the same basic code reader. So we have the universal ability to swap codes and to read other codes. All right, this has been used as one of the evidences of the unity of all living things, right? Because all living things share this code. A simple explanation for why all organisms share this code would be that, as I said before, maybe the original common ancestor of all living things today used a particular code, this particular code, and then it shared it with its offspring, which then shared it with its offspring, and so forth, and so shares that particular genetic code with all of its descendants. And if you imagine that this is a code that's going to be used to make thousands of proteins, and some proteins have thousands of amino acids, which is why you have thousands of pieces of code in every single gene, it would be hard to change this code. If you were to say, okay, I'm going to have another organism, and I'm just going to, when one organism gives birth to another, and how about you just use a different genetic code? It's not that easy, because if you change any one of these base pairs, if I change CUU and I say, now it's going to code for proline instead, right? I'm going to make this PRO. You're going to have this mutation. That's going to be a lethal mutation. And it's lethal because for you and I, we probably have thousands of locations in thousands of genes. All right, well, let's just say tens of thousands of locations in our genes in which you have a CUU codon, and it needs to be read as a leucine in order to make a proper protein for it to function and you to do the metabolism you need to do to stay alive. If you were to replace all of those CUUs and translate them as prolines, you would disrupt thousands of different proteins, and it would be unsurvivable, right? That would be a lethal change. And so once the genetic code is established and is used in one particular individual, any living thing that comes from that particular individual, any offspring, will have to use the same code in order to survive. It's just very difficult to change the code. And so that's been a powerful argument for the unity of all living things, and that all living things share a common ancestor. So now, if we go back to the question, some would claim that the near universal genetic code is not evidence for common descent, right? As I said, many people would use it as evidence for common descent, but there are some who would say that isn't necessarily evidence for common descent. Now, I'll quibble with this in the sense that it certainly can be used as evidence for common descent. That wouldn't necessarily mean that common descent is in fact true, but it is certainly a piece of evidence that supports that particular view. And so to the alternative view is really saying, well, what's another possible explanation for what appears to support common descent? And that other alternative explanation is that if there was a common designer that made all organisms separately, so let's say the God of the Bible, all right? And we would say that we were taking an interpretation that God created all the different kinds of organisms separately from one another, and so they don't have any common ancestry through any physical ancestor. So, I'm saying physical because they, in a sense, have a common ancestor in God's mind, right? And that's kind of the idea here, is that in God's mind, he's creating bacteria, plants, and so forth in a very short period time span, and he uses the same core principles for all living things. And the genetic code is one of the most core things there are, right? And so maybe God uses that as a pattern, right? He's using it for, and then the difference between different organisms is, okay, well, we're using the same code reader, but I'm going to give you different sequences for genes that make slightly different products, right? You're making the products the same way, but if we look at it, let's go back here. If you were to, like I said, if you were to change a UUC in a gene, and you would have this gene, or you say like, I'm going to have this be A instead. So I change the C and the A. Well, then you're going to have a different amino acid being made there, which then makes a slightly different product, which can make the organism different. And so the argument here is that a common designer says, I'm going to change around the actual codons, like I'm going to string together different codes, A, T, C's, and G's, but I'm going to use the same code reader to read everything. I think an interesting application of this, some interesting things to think about, are things like, two things that come to my mind. One is, what if God had wanted to make humans obviously different? All right, like make it completely obvious that they can't have a common ancestor with any other form of life on Earth, specifically chimpanzees. But if you wanted to make that very clear to scientists, he could have simply given human beings a completely different genetic code. All right, so your code, human beings, we use a genetic code, but maybe all of these codes are mapped to different amino acids. And so maybe we make the same proteins, but we have to use completely different sequences to do that because you've mixed up the code, right? So giving a different code would then make chimpanzees and humans incompatible, and you wouldn't be able to find that they would have a common ancestor because there's no way that the same sequence could ever become two different sequences that are read with completely different codes. And so this is kind of reversing the idea of common ancestry, right? If different organisms had very different codes, it would be crystal clear that they didn't, or at least it would be strongly suggestive of the fact that organisms don't have common ancestry, because they would look like they're separately created things with their own unique codes. All right, so that's one thing that I think is interesting to think about. God could have made humans just different than all other life forms, right? If all other life forms had the universal genetic code, and human beings had, and we were, discover that human beings had a completely different code, then that would clearly set us apart. But that's not what's happened, right? It's very obvious that human beings use the same code as other primates, right? We read our codes the same way. We read our genes in the same manner and fashion. You could think of it again, like I said, we run on the same operating system, right? We're all running whatever the most modern Windows version is versus Apple operating system. So the second thing that I often ponder when I'm thinking about this is, I got to remember the second thing first. The second thing that I often think about, I guess you could say it's a ramification of the fact that the code is nearly universal. And I mentioned it before. The fact that it's universal allows me to take the insulin gene from me, all right, or from any human being, take that particular piece of code out, stick it into a bacteria in such a way that we have the bacteria read that code for us. And when it reads the code for us, it actually constructs an insulin protein that is the same as the insulin protein that is in our bodies that we produce. And that's, in fact, how we actually make so much insulin today. If somebody takes insulin, they're taking human insulin, but it's not actually made by a human being. It's made by a bacteria, but we don't call it bacterial insulin. Bacteria don't natively make insulin at all. We've simply given them our gene. They read that particular code because they use the same universal code reader. So you could say, again, that you could say that supports a universal common ancestry, that there's sort of a mix and match ability for organisms to swap DNA. This also helps the evolutionary process because organisms do actually swap information, and that allows them to gain characteristics over time. On the other hand, you could say maybe this was all part of God's design, right? God intended didn't want to prevent us from being able to swap genes, like with biotechnology, right? We can put genes we like from one plant into another plant to give them new properties, all right? We take genes and put them into different animals in order to give them enhanced properties as well. And so, I know some Christians actually don't want to believe in biotechnology, don't think that biotechnology aren't like that's playing God. It's altering the things that God has made. But on the flip side, if he designed everything with the same code reading, the same operating system, essentially it's almost tacitly saying, I designed everything so you can swap all these different parts and you can be creators yourself in terms of generating new diversity by swapping out pieces of DNA from one to the other. And some of it's life-saving, right? Making insulin inside of a bacteria allows us to help a lot of people out. And if the genetic code of a bacteria was read completely in a different way than in a human being, that would greatly limit or completely exclude us from being able to generate insulin in that fashion. All right, so it's you can see it from both directions, depending on your perspective, right? All right, now the last part of this question says, all right, and now we're really kind of building up to this is all background still, just to build up to, I think is the most interesting part of this question. For example, are there other genetic codes possible, with some being just as, if not more efficient than the standard genetic code? So I suggested that had God as a designer wanted to make it clear that human beings do not have any kind of common ancestry with other organisms on Earth, he could have just given us a different genetic code. But when you could push back and you could say, but maybe the genetic code that we have is like just the only way you can really do it. Like the way God designed the physical parameters of this world and the way chemistry works, this is the code you have to use. Like this is just how it has to be organized. You have to, AAA, the way you link AUU, the enzyme you use to link it together, it really needs to go with isoleucine. Like there's something about the chemistry of isoleucine that interacts with the codon, which is the little tRNA thing that matches up and reads your code. There's a physical limitation there somehow that means that this is the most efficient, best way, the best combination. AUU belongs to isoleucine, and GUU is a valine, is always going to be attached to a valine. And if that is the case, then you could make the argument that the genetic code is simply the absolute best, most fine, I guess you could use the word fine-tuned, right? It is a code that if you made it any other way would be less efficient, and an intelligent designer is going to make the best code, obviously, right? You're not going to start with a substandard code. You're going to make the best code. This also could be argued from an evolutionary perspective, though. From a common ancestry standpoint, you could say that there may have been original life forms that used multiple different codes, but many of those other codes were less efficient, and eventually the individuals that had the particular code that we're using now outcompeted all the other versions of organisms that were using alternative codes. And since those alternative codes weren't as efficient, we ended up with one particular code winning out, and then ultimately their descendants essentially took over the world, and that's why all living things use this particular code today. Now comes the big question. Are there other codes? I mean, are other genetic codes possible? Or is there any physical evidence? Like, do chemists know that this is just the only way it can be done? It's the most efficient way. So is there anything else beside the standard genetic code? Well, by now you should have figured out yes, because I called it the nearly universal genetic code. All right. It's not completely universal. There are organisms alive on this earth today that are not using the complete, what's called the standard genetic code. All right, that's actually, standard is better than saying universal, since it actually isn't universal. So this is going to take a little explaining here. All right, so there are examples a variation in genetic codes. The easiest place to see that is in the mitochondrial DNA. So for example, remember your mitochondrial code, you have a cell. In the cell, you have your nucleus, and you've got 3 billion base pairs of DNA in your nucleus. You got gobs and gobs of DNA on your chromosomes. But you also have a mitochondrion. Usually looks like this. And in your mitochondrion, you have a little circular piece of DNA about 16,000 base pairs in size. It's a little more than 16,000, but let's keep it even. 16,000 base pairs compared to 3 billion. So it's a small genome. But interestingly, that genome, it has genes. They have to be transcribed. They have to be read. It's code that's important for running your energy operation in your cells. And so, for example, in you and I, in mammals, UGA, this particular codon, in the nucleus, in your nucleus, that makes a stop codon. Oh, I didn't mention that before, but one of the codes, actually there's 3 of the codes, tell your protein machine when to stop reading, right? You got to know when your protein's done, right? And so there's a signal in your genetic code for this is the end of my protein. There's a beginning code, and then there's an end code, and that tells you like that's your gene in between there. That's your protein making stuff. Well, it turns out that in your nucleus, you make a stop codon. That is the standard code. Like UGA is a stop codon in almost all organisms. But in your mitochondrion, just in your mitochondrion, in mammals, we make tryptophan, an amino acid, rather than a stop code. It was like, well, how do we do that? Don't we need to have a stop? Well, you have 2 others. So you still can stop your codes, stop your proteins. But it makes tryptophan. So that's not the standard code. And also in fruit flies and also in yeast make tryptophan as well, among other organisms. So that's an example of a non-standard code. AUA makes isoleucine in the standard code, but makes methionine, a different amino acid, in several different mitochondrial codes. But all of these codes down here make leucine, but they also make leucine in mammals, so you're making the same thing in your mitochondria and in your nucleus. But yeast make threonine. All right, so what's going on here? How can we have a code that then has different codes, right? Different reading codes. Didn't I make a big deal about the fact that it would be really hard to change your code? I mean, if you change one of these codes into something else, I talked about that being lethal. Like if I would have thousands of genes that are using that particular code. And if I suddenly changed it, like one of my children suddenly was unable to read that code into a stop codon, and now they're making tryptophan, that's going to mess up a whole bunch of proteins, and it's probably going to be a lethal situation. Natural selection is not going to like that, right? It's going to select away any kind of changes that result in those types of bad mutations. All right. Now, that's not the only cases. I know you can't read this most likely, but what this tree is it's saying you're starting in the middle, you have the standard code. And this is showing all the exceptions. So down here, there are 16 different known exceptions to the genetic code. So UGA, I just showed you, UGA stop. The exception is tryptophan. There's another case where AUA is methionine is isoleucine. And there's another case where UAA is a stop and becomes tryptophan. All right, that's a different stop codon. And so you can go through all these, and these are just places where one amino acid is swapped out for a different amino acid. So these are differences in the genetic code. And then the numbers on the tree represent all the different kinds of organ. And these are all just different organisms. These are a bunch of different bacteria. And these are animals up here. And like I just pointed out, you have an alternative genetic code in your mitochondrial DNA for one of the base pairs, or for one of the codons. And then there are some nuclear codes as well. So in the nucleus, there are a few places where we have alternative codes. So now how do we work this in with, all right, I thought universal common ancestry suggested that all living things should have the same code. All right, that would be a prediction potentially of evolutionary biology, that you might expect that if a universal common ancestor had a particular genetic code and sort of like fixed into place, like now this is the code, and then you had that organism have offspring, it should share that code with all of its offspring. And then it should be shared with all living things. But now we come along, we say, well, but there are exceptions to the rule. So now this raises two issues. One is, okay, so now evolutionary biologists need to have an explanation for why there's exceptions, right? Since that's something that is not necessarily expected on just the first blush, right? First look at it. But also, from the intelligent design perspective, this also needs to be explained as well, right? If the intelligent designer is making all organisms with the same plan, right? And saying like, I'm going to use the same operating system on all living things. They're going to all use the same genetic code to read their genes. But I'm going to pick out a few exceptions. This organism over here, this one over here, like this mitochondrion in this particular organism over here, that particular fungi over there that's going to live in this part of the world, it's going to use a slightly different code. Now, when I say different code, we're not talking about all 64 of the codes being different. We're talking about 63 out of 64 codes are the same, and they have like one place where they differ in their code. But nonetheless, even one place differing in the code, if you have a genome of billions of base pairs, that means you're reading it thousands and thousands of times. That's going to be a really bad situation if you try to change that. So both of these positions, a theistic position, an intelligent design position, and an evolutionary perspective, all right, they need to have an explanation for the exceptions to the rule. So I'm going to give you one possible explanation of what's going on here. There is a pattern, right? And patterns are important in science. And the pattern here is that the organisms, the bacteria, for example, that have alternative genetic codes, they share something in common. Mitochondria share something in common with each other. And then I'm not going to go into the nuclear ones, but there's some interesting things about the nuclear ones, but we won't do that example. I don't have it. I've already running really long. Okay, so here's what some of the bacteria have in common. If you look at the bacteria that use what's called alternative genetic codes, and you ask, what is different about these bacteria compared to your average typical bacteria, like your E. coli living in your stomach, which uses the standard code, not an alternative code. These ones with the red dots down here are all types of bacteria that use alternative codes. They have alternative genetic codes. And what's important here is two things. One is this shows genome size, and the genome size right here is 1 million base pairs. All right, and so most free living bacteria that are able to survive on their own have genomes that are 1 million base pair or larger. In other words, they need about 1 million base pairs to have enough genes to be able to do all the operations they need to survive on their own. There are bacteria in this world, though, that have genomes that are smaller than 1 million base pairs. Here's 500,000 base pairs, and all the way down here we have 100 and some thousand base pairs. The smallest known genome of an organism, all right, living thing. And all of these use non-genetic standard codes. The other thing about them is that they have a different GC content. Now, remember I was saying your DNA is A, C, Gs, and Ts. And if you were to look at how many C's and G's, remember C always pairs with G in your code on the ladder, and then C always pairs with T. A always pairs with T. Yeah, I should know that much more clearly than that. So that's the typical pairing way that these pair. All right, so GC content refers to what's the percentage of G's and C's in your DNA versus your A's and T's. And these organisms, you'll see, have low GCs. So if they only have 30% GC, that means they're 70% A's and T's. Now look back up at the code. I threw the code back up here. If you had very low C's and G's, all right, look at leucine right here. Actually, let's look at valine. All right, let's look at valine right here. if you had very low C's and G's, well, you're going to have a G in your code in order to make a valine. Because the only way you can make a valine is to have a G at the first base position of your codon. And then you have a U. That's fine. That's the T's, right? The T's become U's. And then you have UCAG. Well, the C and the G are probably not likely to be very common in the genome. And there's 4 different ways to make valine. This is called the redundancy of the code. You have 4 ways of making the same thing. And by the way, this is why you can have mutations that have no effect on you, right? You can change a U to a C at that position in one of your genes, and even a really important gene that you can't live without. And you would be like, whatever. It still makes a valine, which means I'm still making the same protein, which means I'm totally fine. That's why, a lot of mutations are neutral. They don't harm you. But back to our point of our little tiny bacteria here. If you're a very small bacteria and you don't have a lot of genes because you're a parasite, that's what these bacteria are. They're endocellular parasites. They live inside of other cells, in which case they can parasitize to get a lot of stuff they need. They don't have to have their own genes. So they're small. They don't have many genes. And they don't have much G's and C's. They mostly have A's and T's. So in that case, GUC in genes, let's say there's only 300 genes, and only 50 of those actually include a GU. You only need a couple thousand valines, all right, in order to live, in order to make the proteins you need. If you only need a couple thousand, right, you can spread those couple thousand over the 4 different codons. And what's likely is you're going to have GUUs and GUAs because there is general pressure on the genome to lose C's and G's, right? That's a whole other story, but there's pressure on these small genomes in parasitic situations to lose their C's and G's. And therefore, they're going to prefer to just hold on to the CUUs and CUA codons. And what if there's a situation that arises where they don't even have a GUC anywhere in their genome? Right? That can happen when you have billions of bacteria of a species, and a couple of them have mutations, and they have mutations that change their C's to U's or their G's to A's. And then all of a sudden it's like, wait a second, I'm not even using GUC at all. What happens if you have a mutation to the enzyme called aminosynthetase? And what happens if you have a mutation to that enzyme and it changes the structure of that enzyme such that GUC no longer pairs very well with valine, but maybe it matches up with, let's say, isoleucine? Well, all of a sudden, you have a situation where GUC now makes isoleucine. So now you've paired the codon with what we would think of as a wrong amino acid in the standard code. But this doesn't harm the organism, because if you're not actually using GUC, you never need to make a protein, right, with the valine in that position, because you don't have a GUC at all. You're not reading it. All right, now follow me here. You've got billions of bacteria, and now maybe you have billions of bacteria of a species that all have this particular code. It's a non-standard code. But it's neutral. It doesn't have any effect on the organism. And then one day there's a mutation, and a GUU is mutated to a GUC. So now in that organism, it actually has a GUC, and it's actually in a protein coding gene, and they start to read it, and they read it as, oh, well, this is a GUC, that's isoleucine. And they plug that into a protein. Now, if it plugs it into the protein in the exact wrong spot in a really important protein, that could be lethal. All right, but there's a lot of proteins that can stand to have a few changes, and it doesn't really affect them drastically. And so the first mutation only is in one gene, one place in the entire genome, out of 500,000 base pairs is reading this isoleucine. And in that particular protein, it's like, whatever, valine, isoleucine, it doesn't change the shape that much, and I can continue to function. Well, from that day forward, you're now using an amino acid that's attached to a different codon. You're using a non-standard code. So what I've described here is the process of, I guess really what I've described is the possible process of evolving a non-standard code. But it requires a situation where you get into a very small genome. You don't use some of the amino acids because they're redundant, so you don't need to use every single one of these codes. And once you get to a situation where you're not using them, then mutations can accidentally recode them into a different amino acid. And once that happens, the organism might start using it again. All right? All of this has been statistically shown to be highly likely in these situations. In fact, you could say this is predictable, that this is what will happen if you get organisms in these particular situations. And so when I said there's a pattern, the pattern is that you find the alternative genetic codes in organisms that clearly have had the opportunity to evolve alternative codons. Think about your mitochondrial DNA, right? Mitochondria in organisms. Mitochondria, I said earlier, are only 16,000 base pairs, right? You only have 30, 40 different important genes there, and you may selectively use some codons, right? The mitochondrial genome also has a bias toward ATCG. And so you might be using two particular amino acids, but not the other ones, not the other codons, which gives an opportunity for swapping that one for something else because it doesn't harm the organism during the swapping process. And so this is how non-standard codes can evolve and are predicted to evolve. And so when we look at the patterns across all living things, where we see non-standard codes is where we think they could happen. All right. So that would be, an evolutionary biologist is going to say, well, that's then consistent with a common ancestor with a genetic code that shares it with its descendants. But some of the descendants find themselves in particular situations where they're not using some of those codes, and therefore they can change them out for different codes, and then later on start using those particular codes. And after that, they now have a non-standard code, which they continue to use going forward. All right, there's another reason I bring this up, and it goes back to our question, right? Are other genetic codes possible, with some being just as, if not more efficient than the standard genetic code? What does non-standard codes actually tell us about the perfection of the genetic code? It tells us that you can survive just fine with other codes. In other words, the standard code is not like it's not necessary, right? It doesn't have to be the code. Each one of these potentially could be something else, right? Where am I at here? All right, CCC is proline, but potentially proline could have been made by any one of the other 64 codons. Now, there's actually quite a bit of literature asking this question, whether there might be certain codons that more efficiently match up with certain amino acids. And so there may be some selection going on there where one is better than another. But there are definitely some places where there's probably no difference, right? They're just effectively neutral. And the fact that there are hundreds of organisms that use non-standard codes and they are just as alive as any other organism and they seem to be doing fine tells us that it is possible to have other codes, right? There are other codes that operate in this world that are as efficient as the standard code. All right, so that suggests that this wasn't a matter of this is the only way it could have been made. So an intelligent designer wasn't like, well, this is really the only way that would best work for all organisms. That can't be the reason There's not a physical limitation in terms of how an intelligent designer would have made the world the way the chemistry works that seems to force us to have a particular standard code. Because it now is apparent you can swap the code around. And we can do a bunch of bioengineering to actually intentionally change the code. And I'm sure those things are being tested right now. I think there might actually be some literature on that, but I'm not aware of it. Yeah, where does that get this? All right, I think we've explored this first question. And so we'll go to question number 2. Given the abundance of codon usage bias in various organismal lineages, can wobbly positions in codons still be useful in phylogenetics as evidence for common ancestry if these regions are not entirely neutral? Now, this is, again, a very interesting question. I don't know if I'm going to be able to explore it to the depth that I'm really going to need to. So I'm going to talk a little bit more simply about this one. Because this one's, if you thought the previous question was complex, this one, it ratchets it up several more levels. So what's fascinating here is this mention of codon usage bias. So we've just been talking about codons, right? So these are all codons, all these little 3 base pair triplets, and the mention of codon bias. So we know if we looked at a genome, so let's say we go into the human genome and we ask, hey, you know what, you've got, let's look at 100 genes. And in 100 genes, you're using, let's say isoleucine, you're using it 1,000 times. Like you need 1,000 leucines to make those 100 genes, right? 100 proteins. And so then you were to ask, out of those 1,000 leucines that you made, you must have had a code that told you here's where the leucine is, right? That's what is the information in your genome. And so you go into the genome and you sequence the DNA. And when you sequence the DNA, you can find out which code was being used to make each leucine. And you find out that, oh, you know what? Actually, you were using this one 600 times, and this one is 200 times, and this one was 150 times. And this one was only 50 times. All right, so 200, 800, 900. Oh, that's only 950. No, 150. Okay, yeah, that's 1,000. It'd help if I wrote it a little more clearly. So there's your 1,000 different usages. So that's called codon bias. All right, you're biased. You're obviously using the CUU more often than you're using CUG to code for leucine, even though they make the same thing. Now, this bias can be created by what we just saw before with the bacteria. You might be in a situation where there's pressure to have more A's and T's or to have more C's and G's. And this is one thing you can compare across all different species and types of organisms. In some groups, there's more CGs, and other groups, there's more ATs. And if you have more ATs, well, then you're probably going to be using CUA and CUU more often. And if you have more C's and G's, you're probably going to use CUC and CUG more often. And so that bias it proliferates across the whole genome and affects how organisms use their codes. And you say, like, well, how does that change over time? Well, that's just mutations, right? Because mutations are happening all the time. Sometimes U's are changed to C, or C's to U's, or C's to A's, or A's to G's, right? But if there's some kind of selectional pressure in the environment or in the genome that says, you know what, T's and A's are we prefer that in our genome. Every time a C changes to an A, it's more likely to stay an A for a while versus an A changing to a G or an A to a C, right? So over time, what happens is the genome ends up with more and more and more A's and T's. You can't get rid of C's and G's because you got to have that first base pair be a C. Otherwise, you're not going to be able to make leucine. Oh, that was a bad example because you can use UUA. Valine, you got to have a G, right, in order to make valine. So you can't get rid of G's and C's altogether in your genetic code. Okay, so there's codon bias. Right? And you can measure this in different organisms. You can see how they use their codons differently. And so back to the question is, can wobbly positions in the codons because it's usually at this third base position where this bias shows up. You're using this codon versus this other one, and it's the third base position, which is sometimes called the wobble position, because it can wobble. You can read it in multiple different ways and get the same amino acid out of it, and therefore you can have mutations that don't affect your protein. Can they still be used for phylogenetics as evidence for common ancestry? All right. The common ancestry part of this, I'm not sure how that really affects this, but it doesn't actually matter. It can affect phylogenetics, and phylogenetics is using DNA sequence comparisons, in this case, to try to identify and understand how species are related to each other, or families are related to each other, or individuals are related to one another, right, by common ancestry, right? I compare my sequence with the same sequence in you for a gene, and if we are exactly the same, then it very well may be that we share a fairly recent common ancestor. We don't have to go back too far in our history to find somebody who shares the exact same sequence with us. But if my mitochondrial genome has 100 differences from your mitochondrial genome, then it's likely that we're going to have to go farther back in time to find the common ancestor between us, because you and I are related by a common ancestor, but because at some point there was somebody, specifically a woman, who had a mitochondrial genome with one particular sequence, and she had offspring, and you and I are the descendants of those offspring, but we have different sequences. So all those differences are the result of mutations that have built up over time. So we use those DNA sequences in order to figure out our ancestry, just like we can use DNA sequences to figure out ancestry of species to each other based on how similar they are. And the question here is kind of getting at, if there are biases in these things, doesn't that throw off your calculations? Well, it can, but in most groups, codon usage bias is kind of is fairly uniform among, like, say, all the primates probably have a very similar codon usage bias. So if you're looking at relationships of within primates, you're probably not going to be affected by this. And also, I have to say that all data sets are affected by some kind of bias, right? Where it's not neutral. Characters are, many characters aren't neutral, especially morphological characters, external features that we usually look at and use to compare what we think are most similar to each other. And yes, we can be fooled by that, right? A sugar glider and a flying squirrel look really similar to each other, and they have a similar habitat, and they're nocturnal, and there's a bunch of similar features. And you might be tricked into thinking, because they're similar to each other, there's a bias that those particular features are similar. We might think that they're very closely related organisms when really they're not, because a sugar glider is a marsupial and a flying squirrel is a placental mammal. So their internal anatomy and physiology is very different from each other. And so you just have to be aware of those biases. And anyone who studies DNA is also aware. They have programs to figure out, is there bias in the sequence? More A's and T's, more C's and G's? Because if two groups have a pressure toward getting C's and G's, but they're not actually related to each other, right? If one group of bacteria like E. coli and some other completely different bacteria both have very high GC content, they're going to share a lot of similarities in their sequence. It's going to make them look like they're really similar, even though they don't share a common ancestor to each other. So yes, it can have effect on phylogenetic calculations. But like I said, most phylogeneticists are very aware of those possible problems, and there are ways to accommodate those problems. It's like we all need to be aware of what our biases are so that we can try to avoid and avoid those biases. Yeah, I think I had some other things I was going to say about that, but this mailbox session is getting kind of long, so let's move on to the last question. So all three of these questions came from the same person, so I thought I might as well go answer all three, but my answer to the third question is going to be quite short because I'm no expert in this area. What are the main religions in the world today within both the monotheistic and polytheistic categories? In addition, how common is the acceptance of evolution between these various religions? And I think the second part of this is really the primary question here. So I'm not an expert in the world of religions. All right. I am a very much a follower of the Judeo-Christian viewpoint. Right. And I hate to, in these days, hate to use the word evangelical, but I guess I fit into the evangelical Protestant category most accurately. And but this particular chart, which I just grabbed really quickly, I think gives an overall broad picture to this, right? That that major groups of world religions are Buddhist, Hindus, Jews, Catholics. I remember Catholic and Orthodox are sort of like the East-West divide in the original Christian church. And then you have Protestants, of which there are mainline Protestants, which are a large group, and then you have your evangelical Protestants. Somebody will call those maybe conservative Protestants or historically conservative Protestants, or maybe Protestants that follow some of the original creeds and so forth. Mormons, Jehovah's Witnesses, and so forth, and Muslims, of course, is actually a very large group. This isn't listed by the relative size of these different groups. This is listed based on their answering a question about human evolution. So the percentage who agree that evolution is the best explanation for the origin of life, and that was the second part of the question, is like, how do other religions view the origin of life, or in this case, maybe common ancestry? This question specifically is about the origin of human life. And so the answers here would suggest that, Jews, Hindus, Buddhists, and so forth are very accepting of human evolution. And you'll see that the Jehovah's Witnesses are actually the least accepting of human evolution. Evangelical Protestants, which include a wide variety of groups, right? Everything from conservative Baptist churches that are more followers of young earth creationists, they're not going to accept any form of human evolution, and so they're going to be on the low end of that. So there's going to be a wide variety in there. I think the thing I always remind people when they see questions like this or see surveys like this is this survey isn't very reflective of the question, really. Is the acceptance of evolution between these various religions? It's hard to ask the question, what do you think of evolution? Because the word evolution means so many different things to so many people. So often it's asked in a more specific way. And so you could ask about that all life comes from a common ancestor, but even a lot of people don't understand really what is involved with that or what that really means. And so often it's, do you believe that evolution might be responsible for the diversity of life? Or in this case, is the best explanation for the origin of human life? And what I would say is that many people will say reflexively no to this particular question, whether they know anything about evolution at all. They're going to say no to human life, but that doesn't necessarily mean that they wouldn't in other contexts find evolution perfectly compatible with many of their other views with respect to all other aspects of evolutionary biology. Dog, a canine can become different kinds of canines in different species and evolve within that particular group or something like that. So it's really hard to gauge this. There's been a lot of surveys, a lot of different people have attempted to sort of grasp what is the acceptance of evolution, but until you have an audience that understands even what that word means and what they're accepting by saying yes or no to that question, you never really can tell, what the true acceptance level is of a concept like that is so difficult to grasp. All right, like I said, this is not any earth-shattering stuff, but I wanted to answer your question because it did come to me in my mailbox. All right, that's it. I think I better quit there. That's a couple questions to my mailbox. Keep them coming, and I'll pick out some things that I think are interesting to respond to, and we'll come back with the mailbox episode number 3 at some point. Thanks for listening. We'll talk to you later. Bye-bye.